antibody. An antibody AB, also known as an immunoglobulin Ig, is a large Y-shaped protein produced mainly by plasma cells that is used by immune system to neutralize pathogens such as pathogenic bacteria and viruses. Antibody structure. Antibody as a biochemical polymer is composed of an amino acid building blocks. Type number and sequence of amino acid in the hypervariable region determine the antigen. The antibodies is composed of two light chains and two heavy chains linked together by disulfide linkage, forming Y-shaped structure. The tail of Y ends with the COO terminus, forming fragment crystalline FC. FC represents the ligand of the antibody to other components of the immune system. The antibody recognizes a unique molecule of the pathogen called an antigen via the fragment antigen binding FAB variable region. Each tip of the Y of an antibody contains a pairtop analogous to a lock that is specific for one articular epitope analogous to a key on an antigen, allowing these two structures to bind together with precision. Using this binding mechanism, an antibody can tag the microbe or an infected cell for attacking by the parts of immune system, or can neutralize its target directly, for example by inhibiting of parts of microbe that is essential for its invasion and survival. Depending on the antigen, the binding may impede the biological process causing the disease or may activate macrophages to destroy the foreign substance. The ability of an antibody to communicate with the other components of the immune system is mediated via its FC region located at the base of the Y, which contain conserved glycosylation site involved in these interactions. The production of antibodies is the main function of the humoral immune system. Antibodies are secreted by differentiated B cells called plasma cells. Antibodies can occur in two physical forms: a soluble form that is secreted from the cell to be free in the blood plasma, and the membrane-bound form that is attached to the surface of the B cell and is referred to as B cell receptor (BCR). The BCR is found only on the surface of B cells and facilitates the activation of these cells and their subsequence differentiation into either antibody factories called plasma cells or memory B cells that will survive in the body and remember the same antigen, so the B cell can respond faster upon future exposure. Upon antigen binding, interaction of the B cell with a T helper cell is necessary for activation of the B cell and therefore antibody generation. Soluble antibodies are released into the blood and the tissue fluid, as well as many secretions to continue to survey for invading microorganisms. Antibodies are glycoproteins belonging to the immunoglobulin superfamily. They constitute most of the gamma globin fraction of the blood proteins. Typically made of basic structural units, each with two large heavy chains and two small light chains. There are several different types of antibody heavy chains that define the 5 different types of crystallizable fragment FC that may be attached to the antigen binding fragment. 5 different types of FC region allow antibodies to be grouped into 5 isotypes. Each FC region of a particular antibody isotype is able to bind to its specific FC receptor, FCR, except for IgD, which is essentially the BCR, thus allowing the antigen-antibody complex to mediate different rules depending on which FCR it binds. The ability of an antibody to bind to its corresponding FCR is further modulated by the structure of the glycan present as conserved site within its FC region. The ability of antibodies to bind to FCRs helps to direct appropriate immune response for each different types of foreign object they encounter. For example, IgE is responsible for an allergic response consisting of mast cell degranulation and histamine release. IgE's fragment antigen binding paratope bind to allergic antigen, for example house dust mite particles, while its Fc region bind to Fc receptor epsilon. The allergen IgE FcR epsilon interaction mediates allergic signal transduction to induce conditions such as asthma. Nanoantibody. A single-domain antibody (SDAB), also known as a nanobody, is an antibody fragment consisting of a single monomeric variable antibody domain. Like a whole antibody, it's able to bind selectively to a specific antigen. With a molecular weight of only 12 to 15 kilodalton, single-domain antibodies are much smaller than common antibodies, 150 to 160 kilodalton, which are composed of two heavy protein chains, and two light chains are even smaller than FAB fragments, 50 kilodalton, one light chain and half heavy chain, and single chain variable fragments, 25 kilodalton, two variable domains, one from a light and one from a heavy chain. The first single-domain antibodies were engineered from heavy-chain antibodies found in camelids. These are called VHH fragments. Cartilaginous fish also has heavy-chain antibodies IgNAR, immunoglobulin new antigen receptor, from which single-domain antibodies called VNAR fragments can be obtained. An alternative approach is to split dimeric variable domains from common immunoglobulin G, IgG, from human to mice into monomers. Although most research into single-domain antibodies is currently based on heavy-chain variable domain, nanobodies derived from light chains have also been shown to bind specifically to target epitopes. Single-domain camelid antibodies have been shown to be just as specific as regular antibody, and in some cases they are more robust, as well they are easily isolated and cultured in vitro in large concentrations. The smaller size and single domain make these antibodies easier to transform into bacterial cells for bulk production, making them ideal for research purposes. Single domain antibodies are being researched for multiple pharmaceutical applications and have potential for use in the treatments of acute coronary syndrome, cancer, and Alzheimer's disease. Applications: 1. Affinity capture regions. 2. Crystallization caverns. 3. Target imaging and immunomodulation. 4. Biosensors. 5. In vivo imaging. 6. Antivenom therapy. 7. Anti-infective agents. 8. Immune-based therapeutics. And 9. Biospecific nanobodies.